Okay, so let's talk about proteins now, our third type of macromolecule. So proteins are very precise in how they're put together. Proteins are made up of what we call a chain of amino acids. So it's a long chain. The bond between one amino acid and its neighbor amino acid is called a peptide bond, right? So whenever we have a long chain of amino acids, we often call that a polypeptide. And that's not referring necessarily to the amino acids, it's referring to the bonds between the amino acids. So it's a long strand of peptide bonds, so it's a polypeptide. Um, these long strands of amino acids, when one binds to another, there's a specific angle that a certain amino acid, let's say like down here, let's say lysine and glycine. When lysine binds to glycine, there's a specific angle that it takes, right? And as you go on down that length of the chain, there's more angles, and so you start getting weird shapes like this, right? Well, these will eventually fold over into a shape that has a specific function. If it's a square shape, it can fit in the square hole. If it's a round shape, it can fit in the round hole, and so on and so on. But if it doesn't form the right shape, if one amino acid is incorrect, the overall structure will be incorrect, and so the function cannot happen. So it's very important with amino acids that this order is correct. What determines this order? Well, your DNA does. Your DNA codes for which amino acid should be next in the chain. And so when we talk about DNA mutations and we talk about things, mutations happening, generally we're talking about the misshapen proteins or just incorrect proteins being made. So a protein's function depends on its structure. If you get the wrong order of amino acids, simply just put the proteins won't work. So how does it happen? Well, we have 4 different levels of structures. The primary structure, which is the order of amino acids in the original chain. Then these will go to the secondary structure once you get pretty much the full chain done. And it's going to form with one of 2 structures. Here we have one. This is the alpha helix. It's like a corkscrew. The other is called a beta sheet, and it's basically just a big flat sheet, right? And those 2 things will hook together in different amounts. A couple different secondary structures, some alpha helixes, some beta sheets will hook together, and they form what's called a tertiary structure. All right, so here's the alpha helix, here's a beta sheet, all right, and they've hooked together to form a tertiary structure. Now at this point, sometimes these may be functional proteins. Like in our blood, we have a chemical called myoglobin, which is used for a couple different things, and it's actually a tertiary structure. It hasn't. It could go one more step and become a quaternary structure, but myoglobin itself is tertiary, and it's just a smaller protein. But it functions, and let's say we keep on going and we get a quaternary structure. Let's take a couple of these tertiaries and hook them together. Here's 1, 2, 3, 4 tertiary structures. Hooking them together, we have now a quaternary structure. This is about as complex as a protein can get. All right, this is pretty complex. But this the quaternary structure, the final product here, is a protein that can go do things in our body, build muscles, turn into an enzyme, and help with chemical reactions. And it's made from tertiary structures. Hemoglobin is another chemical in our blood, and hemoglobin is essentially the protein that is responsible for latching onto oxygen, so that way our blood can transport oxygen. Excuse me. And it's a quaternary structure. Myoglobin is a tertiary, and hemoglobin is a quaternary. But let's imagine if we go back here, what if we had one of these out of order? Well, let's say instead of 2 lysines and 2 glycines, we had 2 lysines and 1 glycine. Well, that means that because the bonding angles between them are all different, we wouldn't start forming the right shape. So could we form the right shaped alpha helix? No, we couldn't. It would be bobbled. It would be slightly off someplace. So would that fit nicely on our tertiary structure? No, it would not. So we couldn't make the tertiary structure. We couldn't make myoglobin. Could those form hemoglobins? Well, no, not if they're all being formed incorrectly. All right, so we wouldn't be able to make this. So if the base level, the primary structure is incorrect, you cannot form anything up. And that's why it's so important that this be in the right order. So I'll give you an example, an easy one. Anybody, when you were born, you probably all know that you drank milk when you were born, right? Well, mammals are one of the only species that continues to drink milk after we are juveniles, or babies, essentially. when you start to get older, a lot of people develop, they lose the ability to be able to break down the sugar that's in milk, and the sugar in milk is called lactose. You become what's called lactose intolerant. Why does that form? Well, there's an enzyme, and enzymes are a type of protein that is formed, and it's called lactase, and its whole job is to break down lactose, which is a sugar. Is an ASE, it's an enzyme, ends in OSE, it's a sugar, and usually lactase breaks down lactose. Remember, scientists are lazy. We enable things in very simple ways. Lactase breaks down lactose. Well, if you can't produce lactase, this order of amino acids has gotten out of order. It's not being produced the right way. The enzyme lactase is not made correctly, so you cannot break down lactose. And that's just a good example. This is a really sad example, but it's worth mentioning. Kind of some extreme examples of what can happen when proteins are not put together the correct way. So there was a young lady, I believe she was a senior in high school, and she started getting sick one day. And they took her to the doctor and found out that some proteins in her brain were being made that were not correct. And the nature of the protein that's being made, it's not able to be broken down very easily, if at all, by the other enzymes that are in the surrounding tissue. So what happens is these proteins are being made, they're just building up, and they start taking up space. And inside your brain, there's not a whole lot of room. You get brain matter in there, and you need that. Well, as these things started to grow, they started to squash her brain. And she started losing motor abilities, functionality of her body, speech, recognition, everything else just slowly started to go, and her mom just sat with her and helped take care of her, and within just a few months she died. And it's a very sad story, but she ended up, the disease that she had, or we call that, was called variant Creutzfeldt-Jacobs disease. And it's just an example of what can happen when proteins are shaped incorrectly. Another good example is mad cow disease, bovine spongophoria. Bovine meaning cow, spongophoria meaning spongy, or aka full of holes. So what would happen is that the proteins that were made in the brain of the cows were passed on through a viral infection. And it made what was called a prion. And prions simply broke down a lot of the tissue that were around them, but the prion itself could not be broken down. And so whenever a cow would catch this, it essentially would start going mad because its brain was being broken down by something its body couldn't stop. And we called it mad cow disease. So, all right, so let's continue on with nucleic acids.